So let's take a closer look at amphibian metamorphosis, and then we'll go to insects. So there's two major hormones that are involved in amphibian metamorphosis, and they're derived from the thyroid. The first is called thyroxine, which is shown here. And important to note is that, let me grab my color here. Thyroxine has 1, 2, 3, 4 iodides on it, so it's often called T4. You might see it labeled as T4. The other one, there's a big, if you hear stuff going on in the background, my two dogs are going crazy playing, so just ignore that, as I am. All right, the other one is called triiodothyronine, and this triiodo just basically means that it's got, instead of the 4 iodides, it's got 1, whoop, gotta get on that layer there, 1, 2, 3. So the one that was up here is no longer there. And so they are actually, so this is actually then called T3. They're both derived from the amino acid tyrosine, which is shown here. Tyrosine. And if you look, it looks like they basically took 2 tyrosines. So here's a tyrosine sort of root here without the iodides, right, or iodines. Okay, and then the second one is this other ring structure that gets put on. And let's see. Here's another sort of biosynthetic pathway of that. So here's our tyrosine, here's our T4 and our T3, okay? All right, so let's take a look at how they influence metamorphosis. So one of the changes that happens in anurans, which is what frogs are, is changes in the eyes. And there's a number of changes that happen, but I'm going to focus on one in which the change in terms of the position of the eye changes from a bilateral, so eyes on both sides of the head, to ones where the eyes are facing forward so that they can have binocular vision occurs. So let's take a look at that and how that all happens. As you watch this time lapse, I want you to look at the positioning of the eyes of this tadpole as it matures and metamorphoses into a frog. Watch as they move from a side position to a top position and a forward-facing position. So one of the first things that I want to go through in terms of metamorphosis with you is one of the events that happens in amphibian metamorphosis, and in particular what happens with changes to the eyes in response to those thyroid hormones T3 and T4. As sort of a refresher, let's go through what we talked about a few weeks ago in terms of retinal projections when we studied neuron outgrowth. You remember this image where I had a flower that was projected onto retinal cells. And here's my right and left retina. And recall that the positioning of where the image strikes the retina is really important. And then the projections to the remember the optic disc, or that blind spot that we all have, where all those retinal ganglion cells exit the retina, coalesce together to form the optic nerve. And then there is a division of those neurons such that the ones on the nasal, or this, remember here's your nose. It's kind of a small nose, but whatever. So all the ones here in this hemisphere, and again, we're looking at a top view as if we were to sort of take your head and had a hinge. We could just lift off and look straight down. All the ones here towards the nose or the nasal side are going to project to the contralateral side. All the ones on what we call the temporal or the lateral side are going to stay on the ipsilateral or the same side. And then this region here is the optic chiasm. Actually, why don't I just sew all of that? So here's that label that we had from back then. So recall, then this becomes the optic tract. I think that's everything that we talked. And then this goes to a projection that projects to the region of the brain called the lateral geniculate nucleus, or the LGN. And what that helps us do, again, because this nasal part of the right retina has the image of mostly where the stem and the where the leaves are, as does the left temporal, but at a slightly different angle. Both of those come together on this side, on the left side, and the same thing's happening with the flower parts on that side. And that's what helps give us sort of depth perception. And you've ever done those sorts of, I don't know, those, there used to be a fad a couple years back, well, maybe longer than a few years, where they had the stereo images, where you sort of like would cross your eyes and you would have these two slightly different images that would come together to form a 3D image. That's basically what's happening where the images fall on your retina and then project to the brain, and the brain reconstructs that in a sort of a three-dimensional view. But what happens, what I want to talk about is what happens in the juvenile or the tadpole phase of the amphibian. So let me first get rid of all of this. I'll leave my flower there. Because this, honestly, it's pretty freaking amazing. Yeah. Okay, let's take a look. So here is what it would look like. Actually, let me pull this up. Let me get rid of that. I just have sort of I sort of faded out a little bit of an image of a Xenopus tadpole. Let me get rid of the flower. It's a little distracting there. Okay. Actually, let me just, can I bring that? I'll bring it up a little bit and then I'll bring it back down. Okay, so this is that image. It's from the textbook. And you can see that the eyes here are on the sides of the head. And what you're not really seeing, I actually point out a couple other cool features too while we're here. So obviously here's the retinas in the back. Do you know what these are? So those are the olfactory bulbs, and then these are the olfactory projections that will go into, here's the brain, okay? And these other dark things are a lot of like blood vessels and some other things. They also have these melanocytes that cover their entire body. You can't see the neurons here, so don't try to look for them. But what I want to focus on is the projections from the retina to the brain. So let me dim this out again. And now let me bring the retinas up. So in that tadpole, you can see that the retinas are on the sides of the organism, or the tadpole here, as opposed to sort of being what we had before, which was here, right? Like that. So, and if we looked at, and they do this by labeling, so you can label the neurons and then see where they project to, it turns out that 100% of the ones here on the right, all of those RGC cells, or retinal ganglion cells, cross over and project to the opposite side of the brain, and similarly on the other side as well. So it's very, very different than what we just learned about, and that makes sense in some ways because these organisms do not have binocular vision, right, in the sense that they do not have depth perception in the same way. In the adult frog, I'll get rid of that. This is what it looks like, and I'm going to bring this image up a little more too, because I dimmed it. And get rid of, oh, hang on, there we go. Okay, so here's what they look like in just sort of almost completed metamorphosing frog. It's not quite done, but you can see the shift and from where those eyeballs were on the side. Let me pull up that one again, and I'll dim this. Still down to 30. Can we see one on top of the other? Let me bring this one down too. Yeah, we can kind of see it, not the best. And it's not centered properly either. But regardless, you can see that there has been a movement. And that movement is going to go in that direction. Like so. So the movement is going to go from like the side here, this way. And in fact, it'll rotate even a little bit more. So they do have almost full facing or front facing. And that's going to help them to find prey or capture the prey, not just find them. All right. So let's take a look at what the heck is going on in order for this all to happen. All right. It's really, really awesome. So in order for these tadpoles to turn into frogs with binocular vision, we've got to go from this, not just movement of the retinas, but we've got to go to, oh, not that, that to this. So they've got to go and have projections on the ipsilateral side. And it turns out that our old friend that we talked about before, Ephrin B, and you recall Ephrin, I'll show you that in just a minute if you don't remember, but it's involved in routing of those retinal optics at the optic chiasm. And let's take a look in more detail about how that works. So when we looked at this before and looked at sort of the signaling molecules that help sort out the axons from the RGC, it turns out we talked about a class of them called ephrins. And ephrins were expressed here at the chiasm. And in particular, what their role was to inhibit these lateral neurons from crossing over. So that means that they were inhibitory to this red track, but not to the blue track. And what Nakagawa and his team published in that paper I just mentioned, this one, how Ephrin B regulates the ipsilateral routing of retinal axons at the optic chiasm. Turns out that here's what the situation is in the tadpole. And this is very much a situation as other birds and fish that have eyes on the sides of their head. And it turns out that there is no ephrin expressed at the chiasm at this level. So let me just grab my here. Yeah, that's where I am. Okay. So if I was to say, let's grab an orange because that's what they use. So ephrin, and it turns out that it's specific. There's a lag with my pen. And it turns out it's one of the ephrin Bs. It is not expressed. Well, there's a very long lag, so my handwriting looks weird. Not expressed in the tadpole stage. And what that means, remember that Ephrin B2 is normally inhibitory. And we saw that, and I'll pull that little figure up. God, this is really annoying. It's inhibitory, right? And that's what we saw here. So Ephrin B2, I said, was inhibitory. And suddenly, at some point during development, it turns on that Ephrin B, after this sort of contralateral sort of setup is already sort of, the tracks are laid down, all the projections are there. During metamorphosis, an interesting thing happens. So it turns out that suddenly, at some point, it's not the original, so not the retinal ganglion cells that were from this part of the retina. They actually do not reassort themselves and sort of pull back. Those ones that were projecting from this part of the retina die off, and new retinal ganglion axons are created during metamorphosis, and they are repelled by the Ephrin B2 that is now expressed. So let me grab my little orange pen again. So now F is turned on. And that prevents these from crossing over at the chiasm and keeps them on the ipsilateral side. Okay? And so again, these are new retinal ganglion cells are repelled by Ephrin B, sorry. And that keeps them on the ipsilateral side. That's pretty cool, isn't it? So again, that's now what's going to set up that binocular vision, because now you're getting what would have been sort of our lateral image is going to stay on the same side, but we would call eventually when these sort of rotate in. I'm not showing the, remember just a few minutes ago in the lecture I said that these are going to sort of rotate, right? upwards so that they're front facing. I just don't have that drawn here because it's sort of happening as a continuum. But again, as all these changes are happening, the eyeballs themselves are moving position because the head shape is being remodeled. And that's going to then lead to this permanent orientation where you've got the nasal RGB cells that were originally, all of them were laid down originally, but the nasal ones will stay. The new RGB cells that are generated during metamorphosis, and they're signaled by, God, I should have, how did I not say this? So those T3 and T4 are the signals to do this and this, okay? So that's just one of the things that those thyroid hormones will do, is this remodeling. I think that's just pretty darn amazing. Can you imagine, just imagine this poor little life of the tadpole swimming around, okay? And you've got this little tadpole just swimming around, and its vision just must be all, I don't know, messed up, because all of a sudden you're losing part of your vision because certain neurons are being dying off, and then new ones are coming online and my eyeballs are moving. It must be a very, very trying time. We think our adolescence is hard. Good Lord. At least our eyeballs aren't moving around inside our heads. I should also mention that the expression of these ephrins in organisms that have sort of eyes on the sides of their heads, such as most fish and birds, they don't express ephrin at all during their development. So they are very much like the tadpole. I think that's pretty cool in terms of evolution. So you got a tadpole who is sort of an aquatic organism with gills and a tail for locomotion, and then it changes all of that body structure to become a organism that breathes with lungs, has limbs, and so forth.
When the party was nice, the party was bumping. Yippee-yi-yo.